Yes, I'm talking about sorghum, so we're switching to a less toxic plant source. I want to thank at this point the audience just for staying, much appreciated. Okay, so, and I promise I keep it short. So let's talk about sorghum. Sorghum is one of the world's oldest cultivated crops, which I wasn't so much aware of, but it's currently cultivated all around the globus, and apparently the United States is the main producer, just followed by India and Nigeria, and it is indeed the fifth most cultivated cereal after maize, rice, wheat, and barley, according to recent FAO statistics. The nice thing about sorghum is, and this is the reason why it can be also cultivated in the different climate regions, it is that it's very well adapted to low-input agricultural systems. So it's very resistant towards drought and heat conditions, and is grown for feed and for food purpose, so for animal and human consumption, and also used for many industrial processes, alcoholic beverage production, etc. So but it is an important staple food for many millions of people, especially in the food insecure regions, what we talked about today earlier, as well, such as some sub-Saharan countries. So for human nutrition, as a staple food, sorghum provides significant quantities of energy every day: protein, minerals, trace elements, phytochemicals. This is what I'm in particular interested in, to the human diet. However, sorghum requires some water and labor intensive pre-processing in order to make the nutrients available for the human organism. And some very traditional examples here are germination, also malting, soaking, boiling, and fermentation. However, there's not only one type of sorghum available. As you can see on the picture, there's a huge variety of different sorghum cultivars which are grown in different regions, and even within a country, within a small region. And this is mainly based on the coloring of the pericarp, not only but mainly, which is the flavonoid and other polyphenol accumulation. And this is also one classification scheme into the coloring: brown, more blackish, white, and red. And as I said, it's a varying content then in different types of polyphenols, but also tannins. So the aim of our study here was to investigate the impact of more modern processing techniques on some aspects of nutritional quality and metabolite profile in sorghum. So we asked the question: how would microwave-assisted preparation, how would that affect the bioactive content in comparison to the more traditional techniques, just as boiling and fermentation? Would also starch digestibility be affected by that, and maybe trace element bioavailability? Also considering the different pigmentations we can have for sorghum, is there an impact of those, and is this actually depending potentially on the processing as well? So we had a probably very simple experimental design here. Well, I have to mention we sourced some different white and red cultivars through collaboration from Uganda and Nigeria. We obviously purified and milled them, so we made flour out of that. This was subjected to either fermentation, and we use here traditional fermentation, which is basically mixing it with water and reach a specific pH. Then also microwaving and boiling at different times and intensities. The resulting slurries and porridges, they were dried and grinded, so made again into a fine flour, then in order to be extracted for bioactive. So we looked at different aspects: total polyphenols, bioactivity measured by different in vitro assays, and also polyphenol profiles, starch digestibility, and trace element availability, in particular zinc and iron. I'm just going to present you some of the important results we've obtained so far. First of all, the total polyphenol content in sorghum, which we measured, as you might know, by the 4-lean assay, and we did some different optimizing for extraction conditions. So the acidified methanol seemed to be the one that extracted best from this matrices. And as we can see, we've got, well, we had many more samples available. I just picked some very typical ones. On the left side, you see the red pigmented, on the right, so 3 on the right side, white pigmented ones. So what we can see is that it doesn't all look the same. So depending on the, striking is that depending on the pigmentation, as we also would expect, we see higher total polyphenols. So if you look in blue, I have to explain the color. This is the flour, the unprocessed, which we wouldn't consume normally, but just as a reference, we had that as well. Then in red, the boiled, in green, the microwaved, and in purple, the fermented samples. So we can see that depending on, as I said, the pigmentation, left side red, right side white, the values look very different, but also the individual processing, boiling and microwaving largely reduced the total polyphenol content, whereas fermentation did retain those polyphenols. There are some different explanations available for that. But also we can see that our Nigerian red and white, so they were much lower in each category. They showed much lower values, although they were all should all have been from the same harvest period. Similarly, the bioactivity that was measured with a TIAC, so ABTS reducing property in those extracts, we can clearly sort of see that there are some slight differences, but the correlation is very clear. It also largely depends on the pigmentation and also the country of origin, and boiling and microwaving leads again to a massive reduction, so correlates with the total polyphenol values. We also looked at iron and zinc availability in our samples. What we did is we measured the soluble trace elements after digestion of processed and unprocessed flour preparations. And here what we did is we couldn't include the fermentation here. We can see the microwaved and boiled samples in comparison to the unprocessed ones, and we can easily see that the iron availability in percent is lower in the processed samples. However, the availability of iron seems to be higher out of iron in the red sorghum varieties. However, the differences are not so massive, and in some cases the variation is quite high. Zinc looks different. I just bit higher variation, but looks very similar. We also started looking into starch digestibility, surprisingly low in our samples, and we compared the white and red varieties from Nigeria, which we had. For some reason, my explanation is gone. To the left is the light gray, the boiled. The black ones are the microwaved ones. There's not too much, it's significant, but there's not too much difference. But it looks like boiling actually resulted in a higher starch digestibility compared to microwaving. And this could potentially be explained. We looked at this structure. If you see the unprocessed on the left, in the middle the boiled, and then the microwave, so that the granules are kind of dissolved. So this might be an explanation that the different treatments just resulted in the different granuli, and this resulted in different digestibility. To summarize our results, we have observed so far that the total polyphenols and bioactivity, which is related, are largely depending on the origin and pigmentation of our different varieties. Boiling and microwaving reduce both polyphenols and the bioactivity, whereas the fermentation seems to retain these higher polyphenols and the activity. So our initial hypothesis that microwaving could be some of advantage here this cannot be justified. However, this is an ongoing project, so this is just the first data. We are interested in the polyphenol composition of the different cultivars and the variations due to the processing. Of course, in particular, the fermentation. So I hope to learn also some new things tomorrow. There are quite a few talks on fermentation. We use traditional, but we want to standardize that, lactic acid or with bacteria or with yeast, and have a look whether that makes much of a difference. Also starch digestion, there seems to be some important impact also of particle size and fermentation. Also, in general, we are interested in the sorghum polyphenols and glucose metabolism. So anti-diabetic properties in some country. I know my Indian students, they told me that something, they were not quite sure if it's millet or sorghum, is being recommended by the authorities if you have high glucose baseline levels, so some sort of gruel or porridge then to eat. It's actually a red colored one then. But we're also interested, we started a research line on brain proteins and protein hydrolytes, so peptides. Of course, this I haven't done this alone. This project has been done in collaboration with my colleague Caroline Orfila at Leeds University. She's the expert in starch digestion. Nick Watson, University of Nottingham, is the microwaving expert. And Dr. Pierre Ruhanga, I'm not sure if I pronounce it correctly, from Uganda. He helped us very much in sourcing all these different Ugandan samples, and our students as well. And this research was possible with funding from the Africa College and Organization at the University of Leeds. Thank you very much.
Thank you very much for the presentation. Any questions? Yes.
What is interesting, you mentioned a lot of activity for extract from sorghum. And what about antioxidant activity?
Yeah.
Do you check?
Yeah, antioxidant, yeah.
And this parallel?
Yes, we use the TIAG assay, so which is the ABTS radical reduction, but also the DPPH, and it correlates. This was, shall I go back to the? Yeah. This is the result of the TIAG assay, so this is antioxidant property, antioxidant capacity. There are some different loads. Yeah, it correlates very well with the total polyphenols, although it doesn't exactly, but what you would expect, especially during fermentation, is maybe you would also have some release of bound phenolics, polyphenols, which are normally not available, things like that.
Just a short question. So how do you explain the lower amount of total phenolic compounds in microwave with respect to boiling treatment? Is it something related to analytical artifacts when you analyze the phenolic compounds, or could be related to phenolic loss in water? How did you treat in microwave and boiling? So how did the amount of added water and the time of microwave?
Yeah, there was exactly the same amount of sample and water, and the samples were also contained, so there was no loss, for example, of boiled samples or anything. So there was no evaporation loss. So we treated them the same way. But okay, the ones were boiled, the other ones were microwaved. So the result is a porridgey. It's a bit different to oat porridge. Yeah.
So microwave was stronger treatment, and they relate other amounts of polyphenols?
Pardon? No, we didn't see a difference. We were hoping that microwaving would be of advantage, but it wasn't. It was similar to similar to the boiling, yes. But boiling is not so preferred, I have to mention that, for sorghum, because boiling in particular reduces the protein quality, because it makes protein less available due to this caffeine fraction. So we hope that microwaving will be better. Sorry, you had another question.
I can help your explanation, because in my opinion the most important antioxidant in sorghum are condensed tannins, and they are good dissolve in water.
You mean what? The
Condensed tannins.
so tannins,
Yeah.
yes.
And
yes.
they are very polar compounds, and when you use boiling water, they are removed from plant material.
But we didn't have any loss from the boiling. There can be no loss of the evaporating because it was enclosed.
No, but you are boiling,
Yeah.
and a part of phenolic compounds go to water,
Yeah.
and then I think they separate water and plant material. No.
No, it's like a paste.
Okay.
And you dry it completely, and then you grind it again
okay.
for the analysis. Yeah.
Okay, but
Yeah, no.
I'm a bit now. When you use methanol for extraction for a plant material,
Yeah.
it's not sufficient polarity to extract condensed tannins.
Yeah.
This, and I suggest for the future research
Yeah.
to use acetone. 80% of acetone with
What percentage?
80% of acetone.
Yeah, we tried some
Yes,
different.
this is-
yeah,
this-
we tried acetone
Yeah.
as well. I think it was 70 or 80%, and the HCl methanol was superior. So we tried it with the different sorghum types and different extraction conditions, and that gave the highest results if you base it on that.
But these conditions are the best for condensed tannins.
Yeah. Yeah, I
And-
know. I
and
mean, it depends what you want to
'Cause
measure.
I was
what
working
you
on sorghum
extracted
also,
in it.
and I found this is the best, absolutely. But
Yeah.
of course, depend on the cultivar, because there are some cultivar with small amounts of tannins.
Yes, I know. I think the brown
Yeah,
ones have
yeah.
particular high tannin
In,
content.
in general, Caro with tannins, Caro less, free of tannins.
Yeah. Thank you very much.
Thank you very much.